Hi everyone, I'm Sanel and today I would like to talk about our Spinsolve NMR software and how easy it is to measure multiple samples with multiple nuclei without any user interference. So on these pictures here you can see in terms of hardware how our Spinsolve Benchtop NMRs look like, and you can see in which ways they can be operated with. So on the left hand side, for example, there is this hyphenation with an HPLC system where one of our customers performed online measurements. On the right and middle side here you see a benchtop system placed on a regular lab bench for offline analysis here, or on the right side you see a picture of a Spinsolve system equipped with an autosampler. So on this slide you see a screenshot of the opened queue within your Spinsolve software, and if an autosampler is available, the queue can be equipped here with up to 20 samples. So you see an autosampler here in action at the top right corner, and even without an autosampler, all functionalities of the queue are also available for just one sample. In terms of size, the autosampler is very compact and fits on top of the Spinsolve system, so you can place it in a regular fume hood even if there is an autosampler on top of it. Every Spinsolve system comes pre-calibrated with the nuclei of choice that you can see here at this top row. That means that there is no need for any user interference while measuring the different nuclei. For our demonstration today, we have chosen 3 different samples placed within the spots here 4, 5 and 6. So these samples consist of a fluorophosphonate, a brine sample and a polysiloxane. The goal of this demonstration is to show you how quickly and reliable the Spinsolve systems measure multiple nuclei of several samples in just a few minutes without any interference. Once you enter the name of the sample and you pick the NMR protocols of choice, you just load them into the queue by one mouse click. One can also save a full protocol list chosen for future measurements that don't always have to start from scratch for designing the protocol list. So the parameters of operation can always be modified even while a measurement is run at the moment. Furthermore, the queue can also be modified in terms of sample measurements order. If an urgent measurement, for example, comes up, one can load the sample and always measure it next. Just drag and drop it at the desired position within the queue. Once the queue is started, the Spinsolve software will calculate the time needed for each measurement as well as the time to run the full queue. If an NMR dataset is recorded, one can always open the dataset and have a look into the data while the queue continuously runs further. During the measurements, every NMR spectrum will be visible on the screen. I will now start the recorded video sequence, and our autosampler here at the top right corner will start picking one sample after the other and start the NMR experiments. If additional processing software is installed on the same computer, one can check a box within the Spinsolve settings and directly transfer the data set into an additional software of choice. So now you see that I started the recording. The autosampler is now moving to the desired position, which is sample number 4. Sample number 4 gets marked with the orange color, and the arm moves down with the sample. After the arm moved down with the sample, the measurement time starts. And you see the first protocol recorded, which is in that case a proton-carbon decoupled sequence with only one scan and 7 seconds. Once this is finished, you will see that directly the next protocol will start, which is a proton NMR fluorine decoupled. This was also recorded with one scan, very quickly, 7 seconds. The last proton measurement consists of a proton phosphorus decoupling experiment. So here again, only one scan is needed, 7 seconds repetition time, and you have your spectrum very, very quickly. So the next nucleus of choice was fluorine. So here, fluorine is a very nice NMR nucleus because it relaxes quite quickly. So here the repetition time was only 5 seconds. The next nucleus of choice was phosphorus. For that, we chose 4 scans with a repetition time of 5 seconds, which gave us a total time of 20 seconds for the phosphorus experiment. And last but not least, the carbon measurement for this sample number 4 is starting right now. So here we picked 16 scans with a repetition time of 3 seconds, which gave us a total amount of 48 seconds. And you see how quickly here the signal to noise ratio is building up, and the signals of the sample are coming out quite nicely. Once the measurement of the carbon is finished, like now, the autosampler will again move back to its previous position and release sample number 4. Sample number 4 will be marked here with a green color now, which means that the sample is finished. And the autosampler will automatically move to sample number 5, which is in our case the brine solution. And for the brine solution, we picked a lithium and sodium measurement, which will start in just a few seconds. So here you see the lithium measurement, which only consists of 2 scans with a repetition time of 7 seconds, so a total time of 14 seconds. And you see a very, very nice signal to noise ratio and the desired signal. The next one coming up is the sodium measurement, and this is a very, very fast measurement. You see that here the repetition time was only 500 milliseconds with 4 scans. So in total you have 2 seconds measurement time for this experiment, and then the autosampler is releasing sample number 5 now. Again, it turns green, it rotates to position number 6, and our last sample will be placed into the Spinsolve system. So here for the polysiloxane, we have chosen to show you the measurement for the silicon NMR. So here you see we picked a pulse angle of 24, we picked the number of scans being 8, and a repetition time of 10 seconds, which give us slightly a measurement time above 1 minute. And here you already see the backbone signals of this polymer coming up here at about -40 and -35 ppm. And you can see the monomer signal here building up at around 0 or +5 ppm. The big advantage of our data set is that while you are measuring the data in the bag, you can always transfer it directly. You see here the buttons for 2 additional software packages. So if they are installed on the same computer, our Spinsolve can transfer the data set directly to the additional NMR software, and you are free to use any NMR software you would like for operating or processing the NMR data generated by the Spinsolve system. So we are nearly there. You see all of the 3 different signals are clearly visible. And we have chosen we have showed how easy it is to switch between 3 different samples and 7 nuclei in a total measurement time of 6 minutes. So that was it. Thanks a lot for your time and attention. If you need any further information, please contact us, or if you have any questions left, please reach out to us. You can also find a lot of information online within our blog post here, which is frequently updated, or you can also find us on social media. There we also frequently update our actions and applications that we publish. So thanks a lot and have a nice day.